Hello everybody, my name is Napat Emdee. Today, I would like to present a seminar in topic, Bone, Blood and Brain. How does bone play a role in age-related cognitive impairment? This presentation is based on research articles of Villeda et al. 2014 and Quimian et al. 2017, and my advisor is Assistant Professor Sukanya Jaroenporn. Let's begin with the introduction. Today, we are going to the aging society. People are getting older and older than before. Look at this graph. You will see that when people get older, their score of long-term memory will decrease. We call this memory loss the age-related cognitive impairment. This is really a big problem in neuroscience. How does aging induce a memory loss, and how do we repair it? In the field of aging biology, there is a famous surgery technique called parabiosis surgery. In this technique, researchers will cut the abdominal wall of two mice, and the body of the mice are connected. So, their circulatory system also connected too. Researchers usually connected the body of aged mouse to the body of young mouse. Previous study reported that the blood from young mouse can cross to the body of aged mouse and can rejuvenate many organs in the body of aged mouse, such as muscle, spinal cord, brain, liver, and heart. So, parabiosis is a useful tool to study the difference between young mouse and the aged mouse. This information led Villeda et al. 2014 to their research question: could the blood from young mice repair age-related memory loss in the aged mice? To answer this question, Villeda et al. Did a parabiosis surgery on two groups of mice: isochronic parabiosis group and heterochronic parabiosis group. In isochronic group, the body and the circulatory system of 18-month-old mice are connected together. So, this is a group of aged mice and aged mice, a control group. In the other hand, in heterochronic group, the body and circulatory system of the 18-month-old mice are connected with 3-month-old mice. This is a group of aged mice and young mice. The aged mice in this group receive the blood from young mice. Then, researchers kept two groups of mice for 5 weeks. To study the memory, researchers focused on the hippocampus, the brain region that responsible for converting short-term memory to long-term memory. They want to study, does the blood from young mice share total RNA or transcriptome in hippocampus of the aged mice? And how does researchers study total RNA share? They use microarray analysis. This is a microarray sheet. This sheet contains several wells. And there is a segment from gene of my genome attached to the bottom of each well. For example, there is a segment of gene A from my genome attached to the bottom of the first well. There is a segment of gene B from my genome attached to the bottom of the second well, and so on. So this means that microarray sheet is a collection of my genome sequence. To use microarray sheet, researchers collected hippocampus from the aged mice from both isochronic group and heterochronic group. Then the total RNA were extracted from the hippocampus. Next, RNA were converted to CNA and cRNA respectively. After that, cRNA were labeled with fluorescent tag. and the result is fluorescent probe. Researchers label fluorescent probe from each group of mice in different colors. For example, fluorescent probe from isochronic H mice were labeled with green color, and fluorescent probe from heterochronic H mice were labeled with red color. Then, fluorescent probe from both groups of mice were mixed together. Then, researchers add the mixture to the microarray chip. And this is the example of the result. How to analyze it? Look at the first cell, the value of gene A. You will see that the fluorescent signal is red. This means that the fluorescent probe from heterochronic H mice bind to gene A segment more than fluorescent probe from isochronic H mice. So, heterochronic H mice express gene A more than isochronic H mice. Next, in the second valve, the valve of gene B, the fluorescent signal is green, right? This means that fluorescent probe from isochronic H mice bind to the segment of gene B more than fluorescent probe from heterochronic H mice. So it means that isochronic H mice express gene B more than heterochronic H mice. And in the last one, the fourth valve, the valve of gene D, fluorescent signal is yellow. This means that fluorescent probe from one group of mice attached to segment of gene D equally. So we can interpret that isochronic H mice and heterochronic H mice express gene D equally too. And this is the real result. Researchers found many differentially expressed genes between two groups of mice, and they show this differentiation, this difference in the manner of heat map. This is the heat map. How to read the heat map? The row of heat map shows the list of differentially expressed genes between isochronic and heterochronic H mice. And the column of heat map show different groups of mice. Gray column show isochronic group, a group of a control. And heterochronic group are represented by red column. This group of mice is the H mice that receive the blood from young mice. Yellow color show high level of gene expression. Blue color show low level of gene expression. You will see that in the upper part of the heat map, heterochronic H mice that receive the blood from young mice express this gene less than isochronic H mice. However, in the lower part of the heat map, heterochronic H mice will receive the blood from young mice express this gene higher than the isochronic H mice. Not only the difference in gene expression, researchers also analyze the interaction between these genes. They found many interactions. It is very complicated. But you will see that in the center of interaction, it is the CREB pathway or cyclic AMP response element binding protein pathway. Yeah, researchers found that the blood from young mice affect CREB pathway in hippocampus of H mice. This pathway were reported to related with synaptic plasticity. Synaptic plasticity is the change in synaptic transmission state, which is related to the memory. But this is just a result from RNA level. Researchers want to confirm the synaptic plasticity in higher level, such as protein level or functional level. So the next research question is, could the blood from young mice affect synaptic plasticity of H mice in higher level? To confirm synaptic plasticity, researchers did parabiosis surgery on two groups of mice again, isochronic group and heterochronic group. And they collected hippocampus from each mice from each group. Next, the baseline were prepared from their hippocampus and were tested with 3 different techniques. The first technique is immunohistochemistry. Researchers used this technique to study the expression in protein level of the protein that related to CIV pathway and synaptic plasticity. The second test is Golgi staining. This technique can reveal the morphology of the neuron in hippocampus. So the researcher can count the dendritic spine of the neuron, which is an indicator of synaptic plasticity too. And the third test is extracellular electrophysiology. With this technique, researchers can record neural activity of the neuron. Let's start with immunohistochemistry test. In this test, researchers focus on 3 different proteins that related to CIV pathway and synaptic plasticity: EGR1, c-Fos, and p-Creb. They found that there is a significant difference between both groups of mice, especially in dentate gyrus region of hippocampus. Researchers found that in dentate gyrus region, heterochronic age mice express EGR1 protein, c-Fos protein, and p-Creb protein higher than isochronic age mice. And this means that, the blood from young mice can enhance the expression of protein level of this protein, which is related to synaptic plasticity. The next test is Golgi staining. This is the result of Golgi staining. You will see that this is the neuron in hippocampus, and this is the dendritic spine. This is the dendrite, and this is the dendritic spine. Results show that in dentate gyrus region, heterochronic age mice show high number of dendritic spine when compared with isochronic age mice. So it means that the blood from young mice can boost the synaptic plasticity of the age mice. And this is the last experiment, electrophysiological recording. Researchers stimulate the neuron in hippocampus with electricity at the 0 minute. And they found that hippocampus of heterochronic age mice have long-lasting activity when compared with isochronic age mice. So it means that there is a long-term potentiation or LTP in heterochronic age mice. What is LTP? LTP is a strong synaptic communication, which is related to memory. So with immunohistochemistry technique, Golgi staining technique, and electrophysiological recording, researchers can say that the blood from young mice exhibit a potential to the synaptic plasticity of age mice. And the last question of the research is, how about the behavior of the mice? Could the blood from young mice enhance memory and learning behavior of the age mice? To answer this question, researchers used 18-month-old mice, the age mice. They injected age mice plasma or young mice plasma to this age mice every 3 days for 24 days. Then the age mice are tested with 2 different behavioral tests. The first one is fear conditioning test, and the second one is radial arm Waterman's test, or RAWM test. Let's begin with fear conditioning test. This test contains 2 days, day 1 and day 2. In day 1, researchers place the mice inside the box, the blue box. After that, the mice will receive a toy. Then they will receive electric shock at their foot. They will receive the tone and electric shock for 2 times. Electric shock will stimulate the fear in their memory. And researchers will put the mice out of the box. In day 2, the mice were tested with 2 different conditions, contextual test and tone cue test. In contextual test, the researcher put the mice inside the box, the same box as in day 1. Their environment is the same, so it is the same in context. If the mice can remember this context, they will freeze their body, and their freezing time percentage is an indicator of hippocampus-dependent memory. And in the second condition, the mice were placed in different boxes, the yellow one, but they will receive the same tone as in day 1. Again, if the mice could remember this tone, they will freeze their body, and their freezing time percentage is an indicator of amygdala-dependent memory. Amygdala is another brain region that responsible for memory too, but they are the other region, not hippocampus. And this is the result of fear conditioning test. There is a result from both contextual test and tone cue test. In contextual test, which is related to hippocampus, researchers found that young plasma injection can boost the memory of the aged mice when compared with age plasma injection. But in tone cue test, there is no significant difference between 2 groups of mice. This means that the blood from young mice can boost the memory of the aged mice, which is the hippocampal-dependent memory. The next test is radial arm water mass test, or RAWM test. In this test, researchers will release the mouse in the swimming pool. This swimming pool contains 6 arms, 1, 2, 3, 4, 5, 6, and one of the 6 arms, there is a hidden platform. In normal habit of the mouse, they don't like the water. They will swim, swim, and swim until they find a hidden platform and stand on it. If the mouse swims in the wrong way, researchers will call it as an error. The mouse are treated with this test for 2 days, 5 boxes per day, and 3 trials per box. In the beginning, we can assume that the mice should not remember the position of hidden platform, so they should do many errors. But in the late trial, if the mice can remember the position of hidden platform, they should do less error than the beginning. And this is the result of radial arm water mass test. Results show that the aged mice that receive young plasma do significantly less error than the aged mice that receive aged plasma. So it means that the blood from young mice can boost the memory of the aged mice. Not only these 2 experiments, this experiment and this experiment, researchers also do an additional experiment. They use fear conditioning test again, but they injected the aged mice with 3 different substances: phosphate buffer saline, young plasma, and young plasma, which is denatured by the heat. They did a contextual fear conditioning test, and they found that the aged mice that receive young plasma have enhanced memory when compared with the aged mice that receive phosphate buffer saline. However, when young plasma are denatured by the heat, it cannot improve the memory of the aged mice. So it means that there is something, some substance, some factor in young mice plasma that can boost the memory of the aged mice. But what is it? Which substance in young mice blood is the brain rejuvenating factor, and where does this factor come from? To answer this question, I have to introduce you the new information. The new information about osteocalcin. Osteocalcin is a peptide hormone secreted by osteoblasts in compact bone tissue. When they are released, they circulate in circulatory system of the mice. Previous study reported that osteocalcin in the blood can cross the blood-brain barrier into central nervous tissue of the mice. And there is also a report that osteocalcin is necessary for hippocampal-dependent memory. In addition, there is also a report that osteocalcin in young mice blood is higher than in aged mice blood, both in female mice and male mice. This information is very interesting because it makes osteocalcin a good candidate for brain rejuvenating factor. Yes, this is the research question of Freeman et al. 2017. Is osteocalcin a brain rejuvenating factor in the young mice blood? To answer this question, Freeman et al. Used 4 different groups of mice: young mice, the 3-month-old mice, aged mice, 16-month-old mice, osteocalcin knockout young mice. They cannot produce their own osteocalcin. And the last group is supplied osteocalcin knockout young mice. They cannot produce their own osteocalcin. However, researchers supply ectopic osteocalcin into their blood. Then, researchers did a plasma transfer in this group of mice. There are donors and recipients, and there are 5 treatments. In first treatment, researchers injected the plasma from young mice to the young mice. In second treatment, researchers injected the plasma from aged mice to the aged mice. In third treatment, researchers injected the plasma from young mice to the aged mice. In fourth treatment, researchers injected the plasma from osteocalcin knockout young mice to the aged mice. And in the last treatment, researchers injected the plasma of supplied osteocalcin knockout young mice to the aged mice. They injected the plasma every 3 days for 8 times. After that, the recipient mice were tested with behavioral tests. The memory test that researchers use is Novel Object Recognition Test, or NOR test. This test contains 2 days of experiment. In first day, researchers placed the mice in the arena. In this arena, there are 2 identical objects, the blue objects. The mice have 10 minutes to observe these objects. After that, researchers put the mice out of the arena. In the second day, the test failed. The mice are put in the arena again, but there is 2 different objects. One object is the same object as in day 1, the blue box. And the second object is a novel object, the new object, the red one. The mice have 15 minutes to observe these objects. If the mice remember that this object is the old object as in day 1, they will take less attention on it, and they will take more attention on the new object. So the mice show long observation time in the new object. Researcher will calculate the discrimination index with this experiment, which is equal to new object observation time minus old object observation time divided by 50. If discrimination index is high, this means that the mice have good memory. But if discrimination index is low, the mice have bad memory. And this is the result of novel object recognition test. You will see that the young mice that receive the blood from young mice have good memory. But the aged mice that receive aged mice plasma have bad memory when compared with the young mice. However, when the plasma of young mice injected to the aged mice, this plasma can boost the memory of the aged mice. But when the young mice are knocked out in osteocalcin gene, they cannot produce osteocalcin. Their plasma cannot improve the memory of aged mice. And in addition, when osteocalcin knocked out young mice are supplied with osteocalcin from the researcher, their plasma can enhance the memory of the aged mice. So it's pretty clear that osteocalcin is the main factor in young mice plasma that can boost the memory of aged mice. But researchers also do the additional experiment. In last experiment, researchers used young mice blood that contain osteocalcin. But in this experiment, they use a pure osteocalcin. Researchers used 3 groups of mice. Young mice, the 3-month-old mice, and the another 2 groups is the aged mice, 16-month-old mice. One of the aged mice received saline, and the other received osteocalcin treatment. One group of mice were implanted with osmotic pump device inside their back, and this device can slowly release saline or osteocalcin into their back. Next, 3 groups of mice were tested with 2 different behavioral tests. The first one is NOR test, and the second is the additional test, Morris-Bottomez test, or MWM test. This is the result from NOR test. NOR test showed that young mice exhibit good memory when compared with aged mice that receive saline. However, when the aged mice are supplied with osteocalcin, their memory will improve in the same level as the young mice. You will see that the result is not significantly different between young mice and the aged mice that receive osteocalcin. The next test is Morris-Bottomez test, or MWM test. In this test, researchers will release the mouse inside the swimming pool, and there is a hidden platform in this swimming pool. Around the swimming pool, there are 4 visual pools: visual pool 1, pool 2, visual pool 3, and visual pool 4. They are different in color and shape. Again, the mice doesn't like the water. They will swim, 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 and swim again until they find a hidden platform and stand on it. And the visual pool will guide the mice to the hidden platform. The mice are tested for 10 days and 3 trials per day. In the beginning, the mice should not remember the position of the hidden platform. So they should take long distance and long escape time to find hidden platform. But if the mice can remember the position of hidden platform in the red trial, they should take shorter distance and shorter escape time when compared with the beginning. And this is the result from Wallis Mortimer test. You will see that when compared between age mice that receive saline with the young mice, the age mice that receive saline have terrible memory. They take longer time to the hidden platform, but young mice take less time. However, when the age mice are supplied with osteocalcin, they exhibit a good memory in the same level as young mice. The green one, yeah. From both NOR test, and Wallis Mortimer test, researchers can confirm that osteocalcin is the main brain rejuvenating factor in young mice that can boost the memory of the aged mice. But there is also another question. What is the mechanism of osteocalcin? Which receptor in hippocampus bind to osteocalcin? Researchers want to identify it. To answer this question, researchers look for a clue from the previous research of Uri et al. 2013. This research reported that they identify osteocalcin receptor as an G protein coupled receptor class C or GPCR class C. And they also reported that osteocalcin prefers to bind in CH region of hippocampus. This is hippocampus tissue, and this is the CH regions. How to identify the GPCR class C in CH region? Researchers use in situ hybridization technique. In this technique, researchers design the riboprobe or radioactive label RNA probe. This probe can bind to specific mRNA of GPCR class C in the tissue of the mice. And when they are hybridized, researchers can measure radioactivity in the tissue of the mice. Researchers use 7 candidates of GPCR riboprobe. These are the candidates. And this is the result of in situ hybridization. Researchers found that different riboprobes bind to different regions in the brain of mouse embryo. However, there is only one riboprobe, the Gpr158 probe, that specifically binds to CH region of hippocampus, as you see in this picture. In addition, researchers also did an immunofluorescence study. They test in hippocampal cell culture, and they used 2 different fluorescent antibodies. The antibody of Gpr158, the red signal, and the antibody of MAP2 protein, the green signal. MAP2 protein is the marker of pyramidal neuron in hippocampus. In this picture, you will see that red signal from Gpr158 and green signal from MAP2 protein are co-localized in the same cell. So we can say that pyramidal cell in hippocampus express Gpr158 in protein level. Now, researchers are sure that Gpr158 are highly expressed at CH3 region of hippocampus in both RNA level and protein level. But they are still not sure that is Gpr158 an osteocalcin receptor. So they do additional experiment. They use 2 groups of mice, wild-type mice and Gpr158 knockout mice. Then both groups of mice were treated with saline or osteocalcin in different ways. Then the hippocampus were tested with 3 different techniques. immunofluorescence electrophysiology recording to record the action potential of the neuron. And the third one is novel object recognition test. This is the study in immunofluorescence. In this study, researchers injected saline or ochyocalcin into anterior hippocampus of wild-type mice and Gpr158 knockout mice. And 1 hour after injection, the brain cells are prepared from hippocampus of these mice. Then the fluorescent antibody of c-Fos protein are added to the brain cells. c-Fos is the marker of activated neuron. You will see that injection of ochyocalcin can enhance the expression in protein level of c-Fos protein in hippocampus of wild-type mice, but saline could not. This means that ochyocalcin can stimulate activity of the neuron in hippocampus of wild-type mice. However, in Gpr158 knockout mice, both saline and ochyocalcin cannot boost the activity of neuron in hippocampus. So it means that Gpr158 is an important factor to respond to ochyocalcin. The next study is electrophysiological recording. In this study, researchers isolated the neuron from hippocampus of wild-type mice and Gpr158 knockout mice, and they record the action potential of the neuron by using electric probe. The results show that wild-type mice neuron that receive ochyocalcin exhibit high level of action potential. Ochyocalcin can stimulate the neuron, but the neuron from hippocampus of Gpr158 knockout mice cannot respond to ochyocalcin treatment. It's clear, very clear that Gpr158 is an important factor to respond to ochyocalcin. And in the last study, in OR test, researchers used 3 groups of mice: wild-type mice, Gpr knockout mice that receive saline, and Gpr knockout mice that receive ochyocalcin. In the last 2 groups, they receive saline or ochyocalcin with osmotic pump device that implanted inside the back. Researchers tested these 3 groups of mice with NOR test, and they found that wild-type mice exhibit good memory, but Gpr158 knockout mice that receive saline exhibit bad memory when compared with the wild-type. Although they are just 4 months old mice, the young mice, they have bad memory. However, ochyocalcin treatment cannot boost their memory too. So it means that Gpr158 is very important to respond to ochyocalcin. And yes, researchers can confirm that Gpr158 is an ochyocalcin receptor in hippocampus. The last question of this research is how ochyocalcin triggers hippocampal neuron and induce long-term memory in the molecular level. To answer this question, researchers use focus on BDNF protein. BDNF protein, or brain-derived neurotrophic factor, is an essential mediator of hippocampal-dependent memory. So, researchers hypothesized that ochyocalcin can boost the expression of BDNF in hippocampus of the mice. And to test this hypothesis, researchers used the aged mice, the 16-month-old mice. Then they were treated with pure ochyocalcin, or ochyocalcin from young mice blood. After that, their hippocampus were investigated for BDNF expression. Researchers used 2 techniques to investigate BDNF expression. The first one is RT-qPCR. This technique can reveal the expression of BDNF in RNA level. And the second one is Western blotting. This technique can reveal BDNF expression in protein level. Let's start with RT-qPCR test. In this test, researchers injected aged mice with saline or osteocalcin, and 2 hours later, their hippocampus were analyzed with RT-qPCR technique. This technique showed that osteocalcin injection enhanced the expression of BDNF in RNA level when compared with the injection of saline. The next study is Western blotting. Researchers transferred the plasma from different groups of mice to the aged mice. Results show that the plasma from young mice can boost the expression of BDNF in protein level in hippocampus of aged mice. But the plasma from osteocalcin knockout young mice cannot boost the expression of BDNF in protein level. So, with these 2 experiments, it's clear that the effect of osteocalcin on hippocampus is mediated by BDNF expression, and this is the result of, and this is the discussion of my presentation. I have to remind you about the result from both Verda et al. 2014 and Clement et al. 2017. In Verda et al. 2014, they reported that there is something in young mice that can stimulate CIB pathway in aged mice hippocampus. This factor induces a synaptic plasticity in dentate gyrus region of hippocampus, and they can boost the hippocampal-dependent memory in aged mice. Next, in Clement et al. 2017, they reported that osteocalcin in young mice can enhance the expression of BDNF in hippocampus of the aged mice. Osteocalcin activates the neuron in CAT region of hippocampus, and this region highly express Gpr158, which is an osteocalcin receptor. And the result is in the same way. Osteocalcin can boost hippocampal-dependent memory of the aged mice. In this overall picture, you will think that these 2 research are not related, but actually they are related. Why? Because there is a report that CIB pathway is the upstream pathway of BDNF expression. And there is also a report that not only CAT region that highly express Gpr158 and osteocalcin receptor, dentate gyrus also express Gpr158 too. So we can say that osteocalcin in the research of Clement et al. 2017 is one of the factors in young mice, but in Verda et al. 2014. Yeah, amazing, right? And this is the summary of my presentation. Osteocalcin from bone plays a role in age-related cognitive impairment. For more detail, young mice there is high level of osteocalcin. So this hormone can cross the blood-brain barrier and influence this pathway. So the mice exhibit good memory. However, in aged mice, their level of osteocalcin in the blood is low. So osteocalcin cannot enhance the hippocampus of the mice. And the result is aged mice exhibit bad memory. Yeah, that's it. That is the result. In the last, I have to give a big thanks to my advisor, Assistant Professor Sukanya Jaroenporn, and thank you for your attention.